All right. So today we're going to flip science fair science battle. And so in today's event, you'll hear two different talks by scientists about their research. And after each talk, you'll rate them based on how understandable, cool, or how much you learned about them. And based on your votes, you'll select the winner. Just as a reminder, this is all for fun. And these researchers are working really hard on their science communication skills or how to talk about their research. So your feedback is really important, and it won't hurt anyone's feelings if you don't give them a perfect score. And no matter who loses the battle, everyone's just here for fun. So first we'll hear one talk, followed by some voting, second talk, more voting, and then we'll ask the scientists to answer your questions. And while they're doing that, we'll be tallying all the votes up to announce the final winner. And then we'll let everyone know who won.
I hope everyone's excited for today's science battle because I and Shadow here are both very, very excited for today's event. So, to begin, in corner one, we have Naomi, the Zoomer Lahiri. Naomi hails from Cary, North Carolina, and hasn't left the South since. She is a current student at Tulane University in New Orleans. She loves the classics, and she still plays Pokemon Go. Naomi is always looking for new info to one-up her opponent, and she recently learned that most people only breathe out of one nostril at a time. Who would have thought? I don't know if I'd do it. Anyways, once again, can we give it up for Naomi? She'll be representing epidemiology today. Can we get some cheers for Naomi in the chat? And in corner two, we have Bryan, the flamboyant simulator Bogin. Bryan hails from Cinnaminson, New Jersey, which means Sweetwater in Native American, but there is nothing sweet about Bryan. He may have been raised by Quakers to be peaceful, but he will attack any mosquito within target range. Bryan spends all of his time sliding rocks on ice and sweeping them to a target. Sorry, not all of his time. He does do science, but all of his free time at least. And he is a curling master, and not talking about curling weights, although I'm sure Bryan's strong anyways. And there's only one thing that scares Bryan. He is always respectful to machines in the event that they take over the world. So can we hear it in the chat for Bryan? Okay, and so as you know, for those of you who have come to these science battle events, we will begin with a coin flip. Heather, can you please bring out the coin? How about Naomi? Would you like to call heads or tails?
Sure, I'll call tails.
All right, what's it gonna be? Oh, it is heads. So that means Bryan will be beginning today's science battle. Bryan, do you have any final words to say to Naomi before you begin?
Well, Naomi, I hope you don't mess up because it might go viral.
Ooh, okay. Naomi, do you have anything to say in response to that?
Yeah, I know you're a curler, but right now you're on thin ice because you're going to go down.
Oh, okay. Bringing your curling skills into the fight. Okay. Well, let's fight this out with some science. Bryan, you may begin.
All right. Hi, everyone. My name is Bryan Bogin, and today I will be telling you guys about how bacterial grappling hooks work. Before I jump in, I just want to give a brief background about myself. So I grew up in a small town in New Jersey called Cinnaminson. I went to college at the College of New Jersey, and this is where I began my research journey, and I joined the lab that I have a picture right here. And I'm going to be talking about the research that I did during this time. And currently, I work at Yale University. I'm a graduate student in the biophysics department. And as I mentioned, I love to play curling. And so here is a picture of me sweeping a rock. So now let's jump into bacteria. So grappling hooks help bacteria move to survive. So some of you may not know what a grappling hook is, or if you do, great. But here's a video of Batman pulling out a grappling hook, shooting it, and it grabs onto a pole, and he uses it to escape. So that's an example of a grappling hook. And bacteria use the same principle to kind of navigate their environment. So here's a bacteria cell. This is a bacterial grappling hook, and this is in a representation of food for bacteria. So, of course, bacteria don't eat bread and cheese, but they still need to move to get to nutrients. So these bacterial grappling hooks help bacteria get to food. And so so here's like a microscope image of the bacteria. So there's the bacteria, and here are the pili filaments. And so these grappling hooks are called pili filaments. They're very small, as you can see, but if we take a closer look, we can see it's a very complex system that involves a motor that helps these pili extend and retract and allows the cells to move. So at the end of the pili, there's a grappling hook. And one other remarkable thing about these pili is that they're incredibly strong. They can hold 10,000 times the bacterial body weight. That's the equivalent of a person holding an entire airplane above their head. So that's pretty remarkable. So here's a video of how pili help cells move and kind of what they look like. And here we go. So what we just saw here was first a microscope image of bacteria, and then we zoom in and we see the reconstruction of the bacteria image. So you can see the pili is the green string-looking thing, and it helps the bacteria bind to a surface and then pulls the bacteria towards its location, acting as the grappling hook for the bacteria. So we want to learn more about this process and how they stretch. So no, not this kind of stretching, but this kind of stretching, where we have an elastic stretching, just like a rubber band. So how can we do this? How can we learn about the mechanism? You saw how tiny they look in a microscope. It's got to be very difficult to study these. But it turns out that we have a way to do this, a way to see the atoms and molecules that make up this pili. And in order to do this, we first have to build a model. So we have to construct the model. But first, let's just talk about what is a model. And so a model is a representation of something. So this dinosaur is made out of Legos. It's not a real dinosaur, but it's a model of a dinosaur. And so if you wanted to study this dinosaur, you can now look inside its mouth, like study its teeth, anything you want to do, because it's a model, it's harmless. But a real dinosaur, that could be dangerous to work with, and it's more challenging to study. And so the same principle applies to our bacteria system. So what other models can you think of? And you can take a minute or take a couple seconds and think of any models that you may have used in the past, and you can put that in the chat. And I'll leave a couple seconds for you guys to write some answers. Yep, Legos, a 3D model. Yeah. What about like Minecraft? Does anyone play Minecraft? That's a model. Yeah. All right. Yeah. So you guys know models probably without even thinking about it. So the model, as a lot of you guys said, was Lego blocks, right? So we're going to use an analogy of Lego blocks to represent our pili here. So each of these blobs you can think of as a different Lego block, and the blobs are connected by balls, and they link each blob to the next, or each Lego to the next. And so this pili is composed of different subunits or different Lego blocks. And that's how we build our model. So once we have our model, what do we actually do? We pull them. So the goal is to observe the stretching of the pili because it's a very interesting thing. And we want to, it's something that we can't observe in real life, but we can if we model them. So we're going to stretch them, and this whole simulation is approximately 200 nanoseconds, and a nanosecond is a billionth of a second. So that is a super small amount of time, but in real time, it takes around 12 hours to run. So that's why we need to use something called a supercomputer, which can do millions and millions of calculations at once. So here is the model of our pili stretching. So here it is stretching. And now it's relaxing back to its initial state. So wasn't that cool? That was all on a computer, all run on the supercomputer. So again, we pull the filaments until they break, and we're going to focus on these two states, which are the stretch states, or the transition in this time frame. And remember, 40 nanoseconds is, a nanosecond is a billionth of a second. So that's an incredibly small amount of time. But in the simulation or in the real world, it does take a lot of time to model this. So what can we learn with from our models? So one thing is that as the pili stretch, they actually become stronger, or there's a core in the middle of the pili that becomes tighter and helps the pili stick together over long distances. So back to our Lego block analogy, you can think of that as these Legos have two contacts in between them, but they can undergo a rearrangement where there's now 8 contacts. So we can measure a similar thing for our actual model, and you can see here that as the pili are stretched, the contacts don't go up by just a couple contacts, but hundreds, suggesting that the filament is really coming together, and that core really helps the filament stick together over long distances. So that was really interesting. So what have we learned? That bacteria need food to survive, and they use their pili to get to different nutrients. That they use grappling hooks or pili to do so. And that we can model pili on a computer and run the simulations on a supercomputer. And that we learned that these Lego blocks can undergo a rearrangement, and the filament core is what allows the filament to stretch. So what are the applications of this, and what can we do with this knowledge now? Well, we can design environmentally friendly materials that help the Earth. That's because pili are part of biology. And so if we can mimic their structure and use similar material, that would be environmentally friendly. We can also design stronger and more elastic materials, like as the suit of Elastigirl. So stretchy rubber bands, new tennis strings. What would you make from pili? And again, feel free to comment in the chat, and we can talk about this more. And yeah, thanks for listening.
Thanks so much, Bryan, for such a wonderful talk. You definitely got them excited. Once you mentioned Minecraft, they are showing their expertise in using models. They are very experienced in using models.
Oh, I can tell.
Okay, but if you have any other further questions, perhaps not directly related to Minecraft, but maybe related to the science that Bryan was talking about, please leave them in the Q&A session, and we'll get to Bryan later after Naomi gets to speak, and he'll answer the questions. So once again, use the Q&A feature to ask your questions. But for now, we are going to be rating Bryan's presentation. So you're going to be getting a poll very soon on Zoom that has 3 questions. The first question is, how understandable was the presentation? Were you able to follow what Bryan was saying? And you can vote on a scale of 1 to 5. The second question is, how much did this presentation spark your curiosity and imagination? Did you think, wow, that's cool? Or, were you somewhere not quite cool there yet? And then question 3 is, how much more do you feel like you know about this topic compared to before? And now the science is not yet over. We will now have Naomi, who is going to be giving her talk focused on epidemiology. So Naomi, would you like to begin?
Today, we are going to talk about becoming a disease detective and how to think about outbreak investigations. So that is me up there in the corner. I went to school here at the University of North Carolina, where I studied environmental health and biology. And then now I'm here at Tulane doing my master's, and I'm learning about public health and epidemiology. And this right here, this is my professor, Dr. Susan Hassig, and she taught me everything that I know about outbreak epidemiology and has been a really great teacher to me. I'm sure that you all have teachers that you love and are learning from right now. And she has done a lot of work with COVID recently. And I thought that that was really cool. So even outside of the classroom, she's been talking to different news reporters to talk about how the NBA has changed, how we're celebrating holidays now, and how we should be traveling. So what is epidemiology? So it's the study of people. And this is a little bit of help on how to pronounce it, because I do know it's a big word and can be a new word for some of you. So hopefully that helps to say it. And then getting a little bit more into what it is. So it's looking at science and math to be able to study how disease spreads within a population, all the different factors that can affect it. And so this is a little bit different from just doing something like, say, a doctor would do, because they're just looking at one patient, and we're looking at lots of groups of people, seeing how diseases can affect a bunch of people at the same time. And we want to study this so that we can figure out how to stop them and how we can prevent them and how to control them if there's a large outbreak, lots of disease going on. So that's what we're trying to figure out. And this is us trying to stop the disease. So we're going to look into the 5 W's of epidemiology. So like I said, we're trying to figure out how to stop the disease. In order to do that, we need to be asking lots of questions, just like you do with any other thing in science. So first, we're going to look at who. So this is looking at who is getting sick. In this example, this is someone that's 30 years old. That's a woman, and so she's getting sick. Just something to think about in case there's lots of people maybe in their 30s that are getting sick. Might be something important to look at. Next, we're going to look at what. So what is it, the disease that is spreading? So in this case, we're just looking at the cold. And then looking at how often this could be going on every year. So those are things that we want to be asking. Where is it happening? So in this case, it's happening in Connecticut. And why is it happening? So, and how does it spread? So in this case, you can spread it by coughing. And when is it happening? So like I mentioned, like the flu comes around every single year. Maybe it's only coming in the spring. So those are things that we want to look at. And for this example, they were only sick for about 5 days. So that's the time that they would be sick and possibly could get other people sick too. So next we want to figure out what is an outbreak. So an outbreak can be lots of different kinds of things. And it can look different. So one case would probably be from a completely new disease. So no one has ever seen this before. It's the first time we're seeing it ever. And we want to figure out what's going on there. So one patient in a new place, that would be if there's always a disease that's going on here in the US and all of a sudden we see it here in South America. Well, how did it get there? and how can we stop it? So that's something that we would definitely want to look into, and that's an outbreak as well, because they've never seen it there. Or it can be just there's way more people getting sick than normal. So we definitely want to look into what's going on there. So if there's only one that's normally going on, and all of a sudden we see 3 pop up, that might be a lot of cases for us. And so we want to figure out what's happening, how can we stop it? And these are the things that we want to look for when figuring out, is it an outbreak? So next, we are going to get on our investigation hats to become disease detectives, because I'm going to be sending you back in time to last year to help solve the case of the secret sickness. So this is a situation. It is March 27th, 2020, and the health department in Korea calls you when Tommy, this is Tommy right here, gets sick and goes to the clinic. So they were really healthy until suddenly they had fever, muscle pain, and then we're getting really sleepy and tired. Well, going back to the 5 W's, we're going to be trying to go through a checklist and making sure that we answer all of the questions that we had talked about earlier on in the talk. So just make sure you keep your eyes and ears open for that. But in this case, we figured out where it was. So it's going on in Korea, like I told you right here. And then when is it happening? So the first case that we saw was Tommy, and that happened on March 27th, 2020. So those are things that you want to note down to figure out what's going on, if an outbreak is happening, and we want to keep an eye out in case other people are getting sick. So let's find out more information. Okay, so we start by talking to Tommy, and we figure out what they've been doing before they got sick, so we can figure out where they might have gotten sick. And they think it could have been from Spa A, or it could have been from Job B. Tommy remembers seeing someone cough at Job B, though. So, I don't know. How do you think that they got sick? Was it A, Spa A, B, Job B, or C? Do you think it's too soon to find out? Type A, B, or C in the chat when you have your answer. And I'll give you a little bit of time to do that. Okay, I'm pulling up the answer. So it's C. It's way too soon to tell. We don't have any other information. Just because Tommy thinks that they might have gotten it from Job B doesn't necessarily mean that's where they got it. So we'd want to do more investigating. So next we find out that Tommy, who was 12 years old, was soon joined by Marvin, who was 7, Polly, who was 72, Hannah, who was 30, and Gil, who was 55. And they were all feeling the same kind of sickness. They were feeling really sleepy. As you can see here, they had a fever. They had those muscle pains. So those are all the same things that we saw from Tommy earlier. And as you can see here, there are people that are all different kinds of ages. So it doesn't really seem like it's affecting, one person more than the other person. And so since there's lots of cases here, we haven't really seen it pop up in this place before, we would say there's definitely an outbreak. And going back to our checklist, this is what we can answer so far. So now we've just added on who. So it looks like it's affecting all different ages of people. Where is it? It's still in Korea. And when is it happening? That first case was still March 27th. So next you talk to the doctor to find out more. So the doctor thinks that it is COVID-19. Once you talk to them, they've looked at the symptoms like we talked about earlier. They have muscle aches. They are feeling kind of feverish. And so next, we would want to figure out, okay, what does the doctor do? They'd want to get it tested. So we'd get it tested in the lab. Next, we need to figure out, what does the test say? And it comes back and it's positive. So this, we definitely know, is an outbreak of COVID-19. So let's consult our checklist again. So now I've answered, it's affecting all people, it's COVID-19, it's happening in Korea, and this is the start date. So now we just need to find out why. Why is it happening and how is it spreading? So we need to figure out how they're getting sick. So after we talked to all the patients, we got a list of all of the common places that they went. So most of the places that they went were Spot A, Job B, or Concert C. And so based on the number of people that went to each location, where do you think they most likely got sick? I'm going to ask you on the next slide to vote. So take a look at this before we move on. Okay, we're flipping over. Where do you think they most likely got sick? Make sure you put A, B, or C in the chat whenever you think you have your answer. If you haven't voted yet, make sure you vote, and I'm going to reveal the answer in 3, 2, 1. So it was spot A. So we figured out the most likely place is probably spot A because there were 4 people that got sick, and only 2 people went to job B, and only 2 people went to concert C. And we see that there's 1 person that didn't get sick at all from going to spot A. So what happened there? Let's find out. Where did Hannah get sick? So after we talked to everyone again, we find out that Hannah and Gil were friends and they ended up going to the same concert. So since COVID-19 can spread from coughing and sneezing, this is probably where they got sick. So now we figured out all of the answers to our 5 W's. So what should we do next? After we figure out everything that happened with the 5 W's, we still have some work to do. And so what do you think we should do next? Should we A, close spot A, B, get treatment for all of the patients that are sick, C, test everyone for COVID-19, D, tell everyone on the news so that they know how to stay safe, or E, should we do all of the above? Make sure to put it in the chat. So E, we don't want to do all of the above. Just because we've solved the 5 W's doesn't mean it's over. We still need to make sure that everyone is safe and we're really helping to stop the disease. So we solved the case. For now, spot A is closed. Everyone was tested and 10 more cases were found. And so you were able to help stop the disease. And sometimes it can be a little bit messy and not all cases you figure out what the answer is. So sometimes the disease is still out there, and that's why we need to always have more disease detectives like you to help figure out what is going on. That's our little party hat because we did it. And this is actually based off of a case that happened last year. So someone that's a disease detective, just like us, went and did an outbreak investigation to look at COVID-19 in the community in Korea, and luckily they were able to stop it. And there's lots of different kinds of epidemiologists. So there's not just the kind of case that we looked at where there's outbreaks happening all over the place. You can have people that are doing nutrition, that are looking at the environment to figure out what's happening with other bugs or disease from plants. We can look at how moms might spread disease to their children, and you can look at cancer. So there's people that are doing things in all different areas. And then these are just some fun things to explore that I also looked at when I was in class. So this is the Junior Disease Detectives comic book. So this goes through an outbreak, kind of like we did here, except they put in a lot more information and do a lot of really cool things. And that's from the CDC, or the Centers of Disease Control, which help to stop disease here in the US and also look at diseases all around the world. And then this one is called Solve the Outbreak. So you can kind of do like we did here, answer a lot of questions and really figure out how to solve them. They have 2 different levels and lots of cases that are really cool. Like you can see here, this one is like the Queen's Killer. I've done that one. And that one was pretty awesome. And I'll make sure to put these links in the chat so you don't have to get them now. But yeah, if you wanted to look more into this and you thought it was really cool, a great place to look. And that's the end of my slide.
Thank you so much, Naomi, on helping us all become detectives. I wish I brought a hat so I could join you here, but hopefully they all had hats at home. I know from looking at the chat that even if they didn't have the hats, they definitely were performing like true detectives. They were really great. Now we are also going to do the same thing that we did for Naomi, that we did for Bryan. So we're going to be pulling up a poll, and it has the same 3 questions. The first question is, how understandable was Naomi's presentation? Number 2, how much did this presentation spark your curiosity and imagination? And number 3, how much more do you feel like you know about this topic compared to before? Looks like everyone's just about done. We are going to be closing the polls now. And Matt is going to be joining us now to ask Naomi and Bryan all the questions that you've asked them throughout the event. So Bryan, Naomi, and Matt, come back for the final round of the science battle.
All right, Naomi and Bryan, great talks. So many questions that these students have from your talks. First one, which has had the most upvote so far, is for Bryan, which is wondering how far can these pili grappling hooks go?
So the pili are actually longer than the bacteria themselves, and in like the microscope images that I showed you before, you saw how long they were compared to the bacteria. So they can go like really far, and that motor that I told you about can control how far they go. So it's actually, they can sometimes be shorter, sometimes be longer depending on where they want to go and depending on like where the nutrients is.
Nice, excellent, thank you. All right, next question for Naomi. How do epidemiologists collaborate to track a large worldwide pandemic?
Yeah, that's a really good question because there is a lot of that going on right now. So I didn't get too much into this, but we don't work alone. That's the answer to the question. So there's people like me that are figuring out how is the disease spreading, but then you have to talk to all different kinds of scientists from all over. So there's lots of medical doctors. So they're sending in information, what their patients are looking like, and we get to look at that information. Then there's people that are talking to the public to let them know, hey, this is what you should do. If you think you might have the disease, you need to go to the health department, you need to go to your doctor. So those are also important people on the team. And basically, you talk to a lot of other people, just like we're doing right now. I'm learning a lot from Bryan. And so you just gotta talk to a lot of people, and that's kind of how collaboration goes. You just try and reach out to everyone that you know to figure out what you don't know.
Great, thank you. All right, let's mix in some of these personal questions. So Bryan, one of the students is wondering, do you think you have what it takes to become an Olympic curler?
Oh yeah, sure. I'm training for the Olympics are next year, right? Winter Olympics. Yeah, that's what I'm training for.
Nice, excellent.
Unfortunately, the season was canceled this year due to COVID, so set my goals back a bit, but I'm working on it.
Yeah, I do remember watching the curling back in the last Winter Olympics, or I remember falling asleep while watching the curling because I think it was at like 2 a.m. Naomi, one of the students is wondering where you got that cool hat. Where it is?
That's a really great question. So I'm currently in New Orleans, and we do this big celebration every year called Mardi Gras. So I got it last year at a big celebration, and someone threw it at me, and now I have it.
So it's gonna be pretty hard to get a hat like that one, huh? All right. Now I guess we should get back to some science. So Bryan, for you, would the bacteria die if the pili were surgically removed, or would they be able to grow new ones back?
Yeah, that's a great question. So the pili are important for their ability to grab onto things, like I talked about. So if you take away their pili somehow or prevent them from functioning properly, then the bacteria won't be able to grab onto things. It'll stop them from possibly infecting people. And so that would be a really cool way of making an antibiotic target. So yeah, that's a great point. In terms of like surgically removing them, like I guess if you like ripped one of them off, it could grow a new one because there's a pool of the subunits that it uses to make new ones. But if you got rid of all of them, yeah, it would significantly prevent the bacteria from reaching its food sources. And yeah, so great point.
Nice, thank you. All right, Naomi, the students are wondering if you kind of have an idea of when we could either like eradicate or defeat COVID. Is there kind of an idea?
Also, very important question, and I think a lot of scientists right now are trying to figure out the answer. I wish I could tell you, but yeah, I don't have the answer, and that's what everyone's working on right now. So we're really trying to figure that out, and hopefully soon. But yeah, as you can see here, we just did a small example, and there's a lot that goes on when trying to get rid of something that's spread to so many different kinds of people. So yeah, we'll just have to wait and find out.
Yeah. I guess going off that question, is wearing masks, is that could be a common thing that's going to be new to our everyday lives, or is there going to be a time when that's kind of gone?
Yeah, I think that just depends on how it goes and what we end up doing. I know that in China, they had an outbreak, not COVID, but they had a different kind of outbreak that spread the same way, where if you coughed or sneezed, and then people started wearing masks when they got sick, and that just became a regular thing. So I don't know if that will end up happening here or not. Again, I think it all just depends. It's really hard to know right now. I think you're confused, scientists are confused. Hopefully we'll be able to figure out the answer soon, and definitely hear about it.
Yeah, I think we are all hoping for that to happen. Okay, Brian. Do bacteria have other movement, other methods of movement besides the pili?
Yeah, some bacteria have these things called flagella, which is more like a swimming motion. And so you can think of them, I don't know, like a fish has fins. And so it's more for like propelling itself through fluids. But the bacteria that I work with, or the ones that have pili, actually don't have these flagella. So if you got rid of the pili, it would really be stuck. It wouldn't be able to move very efficiently.
Nice. All right. Naomi, a question for you is one of the students was wondering, what if our disease investigation goes wrong? What if we're not correct after?
Yeah, like what if you end up not getting the right answer?
Right.
Yeah, so sometimes you don't figure out what went wrong, but the best thing that you try to do is just trying to figure out as much information as you know. And from there, try and figure out how to stop people from getting sick. So yeah, sometimes you don't find out the answer and that's really hard, but we do try our best.
Great. All right. I think let's do one more question for each of our speakers. So first for Bryan. So the students just wanted to double back on why do the pili look like grappling hooks? Why?
Yeah, so that's just a analogy or a representation of the pili's function. They don't actually look like a grappling hook. They just look like a kind of string, as you saw in the microscopy images. But their function, they have the ability to grab onto things like a grappling hook, and so it's a convenient representation. And yeah. But they don't actually look like that. They look like the strings that you saw in those images.
Nice. Yeah, I guess, yeah, models can't always be the most accurate, but definitely always very useful. All right, and then I guess or maybe we have time for a few more questions. So Naomi, people were wondering, how do you get information through other people? Like, how do you know who was at the concert? How do you know who was at the spa, kind of?
Yeah, so you have to ask them and do all of these interviews. Just like we're asking questions now, you ask other people lots of questions. Obviously, this depends on like if they're telling the truth. So hopefully they're telling you all the information so that you can have it. But that's another example of how things can go wrong in an investigation if everyone's not telling you everything.
Nice. Okay. I guess I lied earlier. I think we have time for one more question each. And I think this is going to go kind of towards your own researches. So for Bryan, are you or other scientists working on developing more eco-friendly elastic plastics, kind of using these bacterial pili?
Yeah, so my lab is more focused on my lab that I worked in college at least is they're focused on understanding the mechanics of this process and the mechanism of how they stretch, and the application of that is developing these new like environmentally friendly biomaterial. So my lab's not directly working on that, but as Naomi mentioned, science is all about collaborations, and so we're definitely working with people and we're sharing our research so that other researchers can take advantage of this and develop cool new environmentally friendly material.
Yeah, nice. Yeah, collaboration is so important in science. That's something that should be taken away. And then Naomi, people are just wondering what you're currently working on. What is like your project?
Yeah, so currently I'm not doing anything in COVID, but something that I didn't mention is people that are looking at the different effects of COVID. So right now I'm doing research that's talking to different kinds of people, seeing if their mental health is okay after COVID, how's their, like, are they eating properly and things like that. And I'm taking that information and reporting it back to the hospital so the hospitals know how to best help their patients. So that's some of the work that I'm doing right now.
Great. Great. Yeah. Thank you guys for answering all of those questions. The students loved both of your talks. And I will now hand it over to Josie to announce the final results of today's verses.
All right. Okay, so I can tell you from this quick little data workup that I just did that these were both really, really well-loved talks. I think people are really excited by them and definitely learned a lot. So we'll start with a big thank you to both of our speakers. But we should see really how does the scoring pan out. So before I show you the results of all of the polling, I'm going to need everyone to do a quick drum roll on your desks. All right, let's see. So the first question we asked you all was, how understandable was the presentation? We asked you to rank this on a scale from 1 to 5. On average, you all gave Naomi a 4.5. That's really high. You understood what she was talking about. You gave Bryan a 4.2. That's still pretty good. Most of you got almost all of what he was saying. That's fantastic. All right, question 2. How much did this presentation spark your curiosity and imagination? All right, same kind of scoring. You gave Naomi like, what do we think? That's a bit above a 4. That's pretty good. All right, Bryan is right underneath 4. These are still great scores, well above average. Great. Okay. Question 3, how much more do you feel like you know about this topic compared to before? Did you learn some things? Can you go have a conversation with someone else about this now, maybe? Naomi got like about, let's say, a 4.1 for that. All right, Bryan coming in closer to 4.2. Very exciting. All right, so we're going to take an average of all of these points to get you our total winner, our overall winner, who is going to be Naomi. Congratulations to our winner, and thank you to both of our speakers. We have a lot of yays, a lot of people who maybe don't necessarily agree with the winner, but happy either way. All be good sports about it. Okay, so thank you for all the work you guys put in.